Okay, that's good. Okay, and then I'll just name you host. Okay, I think you're the host now. Is it the host on that computer or is it the phone? Just to make sure.
Yeah, I'm the host.
Perfect. Okay, I'm gonna pause this. Okay, are we ready?
Mm-hmm.
All right, so this is our October. Welcome to our October virtual lunch and learn. Stan's gonna teach us some new things today. And this is, I'm recording, so this will be posted on our website probably later today. So if you know of someone who could not make it or someone else who would benefit, please share that. And if you have questions as we move through, if you could just put them in the chat and I will let Stan know so that he can pause and answer your questions. And with that, without further ado, I will hand it over to Stan.
All right, great. Thank you, Ashlyn. Welcome, everybody. I would like to let you know that we'll have this hopefully fairly short little overview of fluorescence, how it works, some hopefully practical and useful hints and advice. And then when we're done, if some of those people who will want to hang on, I will walk over to our fluorescent microscope and I can share a screen there and we can look at some particular things regarding fluorescence and cameras and noise and such. So with that, let's see if I can make this this thing work. But I think Ashlyn, you'll have to let me share the screen. Otherwise, we'll be stuck here. Okay.
Well, I have that you are the host, so I'm not sure.
Oh, good. But,
oh, it's the other one. Oh, hold on. It's your other account. That's
yeah. Enable the other me.
Yes, 1 second.
Yeah, I think that will be the one on top, the Stan Vitha. Up there.
Okay, try that.
Okay, so, all right, I think that will work. Okay, share. All right, can you guys see this slide, the title?
Yes, we can.
All right, great, thank you. So this is the introduction, and in case you haven't, you're not aware of that, this is brought to you from the Microscope and Imaging Center at our great university. And hopefully it will, all right, there we go, next. Okay, so that's our center. So let's see, we will, first I have to tell you a little bit about the MIC, because we do all sorts of things. We can do sample preparation, imaging, image analysis, provide that as a service, as training. We love to talk to people about their microscopy in their projects, if they need help with some troubleshooting, then collaborations and such. And also, there are some formal courses in the spring semester. The Biology 608, which is the optical microscopy course, and new this coming semester is the Biomedical Engineering 689 electron microscopy course. And of course, towards the end of the month, we will have an open house. October 23rd is part 1. And there, I was told that there will be part 2 as well. So there's, yeah, there's a lot of things coming up. So this is just a brief outline, so for you to kind of look at it. And if you needed more information about the theory of fluorescence and fluorescence microscopy, there is a link that will give you too much information on that. Okay, so how does a fluorophore or a fluorescent molecule work? This is where we like to think, or it's useful to think of light as a particle, as a photon. So light hits a molecule, gets absorbed, and the absorption is fairly fast. It's in attoseconds. And that brings that molecule from the ground state to the higher energy state, and there it will lose some of that energy, and then it sits in this excited singlet state until it can emit a photon, and this is where we get fluorescence. But a lot of times we don't get a photon. Maybe the energy is used in a chemical reaction or transferred to other molecule, and unfortunately for us, no signal. So the fluorophores can be cycled in and out of the excited state many times, but not forever. And eventually, the molecule can be destroyed, especially if, when it's in the excited state, if you have oxygen present. That's why a lot of the microscopy mounting media contain some sort of an antioxidant or scavenger of molecular oxygen. For live cells, then you have some special cocktails that contain either the oxygenase enzymes or even just the vitamin C, ascorbic acid. And if you were looking for recipes for some of these mounting media, there is a link here in the bottom. So, fluorochromes or fluorescent dyes. What we like to know about a fluorescent dye is where is the absorption peak, because that will tell us what the wavelength of light we should use for excitation. And hopefully we'll be able to use something fairly close to the absorption peak so that we have the most efficient excitation. And then of course, we look at the emission peak and try to capture or detect as many of these fluorescent photons as possible. Superimposed over this graph is a spectrum of one of the old-fashioned, fairly common light sources, your mercury lamp. And you can see that it's a continuous source, but it also has some fairly strong peaks. A lot of traditional fluorescent dyes were designed to coincide, their absorption to coincide with this strong emission line so that you get a really bright signal. But that's not always the case. So for instance, here in the bluish, greenish part of the spectrum, you don't really get any big peak. So that's why if you go from one microscope with this light source to a different microscope, let's say that has either LEDs or maybe even lasers, you may be surprised that your green signal that was so dim relative to other signals all of a sudden is really bright, or the other way around. So that's your absorption and emission and light source spectrum. So we have all sorts of fluorochromes. Some specimens are naturally autofluorescent, and that's usually a bad thing, but sometimes we can take advantage of it. You know, chlorophyll in plants is, or pigments in eyes, and many other compounds can be autofluorescent. But of course, we have chimeric proteins or genetically encoded probes like green fluorescent proteins and many other colors. And of course, there are the synthetic dyes that are available. Typically, these dyes are much brighter and more photostable than your fluorescent proteins. So some are the traditional fluorochromes like fluorescein or FITC, acridine orange, some other dyes. A lot of these, they're well known, but they are somewhat dim, not as bright. They are not as photostable. Luckily, there are lots and lots of new dyes. Most well known are the Alexa Fluor dyes from what was originally Invitrogen, now Thermo Fisher. And they've been around for a long time, but there are many, many other makers which make very good dyes, and some of them will cost you a lot less money than the Alexa Fluor dyes. So they're brighter, more stable than the old dyes. So if you look in your freezer and see some sort of antibody conjugated with fluorescein, just throw it out and buy something brighter, unless it's something really specialized. So, and for fluorescent proteins, everybody knows the GFP protein, but there are many other colors and also many versions. Some are much brighter than the good old GFP. And if you look here at this link, you can display your fluorescent proteins based on many criteria, whether you want the brightest or the most photostable, or whatever your parameter, you can plot it any way you want. Now with GFP and other fluorescent proteins, if you're imaging cell cultures, certain components in the culture medium can make the protein to be less photostable, or can lead to some really interesting photoconversion reactions that are typically undesirable. So just a word of caution, look at the literature and see if you need to use some sort of a medium that is missing the riboflavin and pyridoxal. That could make your life easier. So let's see if now if this little video will work. So fluorescence excitation, okay, and lifetime. So what this is a short video that was recorded. There is a citation here at about 100 billion frames per second, and I think it's really cool if I can do it. I'll try it once more. Here you go. So a packet of photons hit this cuvette with a fluorescent dye, and these molecules are now excited and sit in the excited state, and the whole ensemble is gradually, one by one, they're emitting photons, and you can see that it can take several nanoseconds before all or most of the proteins or excited molecules go back to the ground state. So the average time that the molecule spends in the excited state, that's called the lifetime. So I think it's a really cool video. So now we know what fluorescence is, and so how is a typical fluorescent microscope designed? So nowadays the most common is so-called epifluorescence, and this is where the excitation light is coming through the objective and to illuminate the sample, and then we also collect the signal, the fluorescent photons of the fluorescent signal also through the objective. So typically you have the light source, you have some sort of a filter so that you can select just the right wavelength for excitation that is matching the absorption peak of the dye. And there is a special dichromatic or dichroic beam splitter. It's designed, in this case, we're using blue light, so it reflects blue light. We illuminate the sample. Lots of photons just go through, but some get absorbed, and lots of them also just are reflected back. We don't want those because we want the much weaker, slightly longer wavelength fluorescent signal. So because it's a longer wavelength, it passes through the filter, and we further clean up the signal by the emission filter so that we suppress as many of the original illuminating light as possible. And then we can either look at it directly or have a camera or some other detector to record the image. So this is epifluorescence. So at the heart of the epifluorescent microscope is this set of 3 filters, and we'll talk about it a little later. So microscopes could be upright. This is our little Leica fluorescent microscope, or inverted, where we're looking at the sample through the bottom, which is good for live samples that you grow in some culture chambers or petri dishes and similar vessels. So, filters. I mentioned that the heart of that setup is this set of 3 filters. They're typically mounted in something called a filter cube. And when you, in your lab, if you have a fluorescent microscope, you know, you'll have something like that. Now the question is if those filters are really any good. So there is an example here on top is an old filter that is no longer made, but you'll find it in many microscopes. And if you look at this particular graph, you can see that this blue is your excitation filter. And so you were exciting with blue, but it only has about 80% transmission. So we lose 20% of your excitation light. And then we have your emission filter. So we're looking at sort of greenish fluorescence in 500 to 525, something like that. And that filter also has only about 80% transmission. So when you take it together, you only get about around 60% intensity that from what is possible. So this new set has much higher transmission for the excitation and emission, and also the dichroic mirror has more transmission. So you get much more signal with a new filter. So when looking at multiple colors, of course, we have to worry about overlap or crosstalk between your different fluorescent channels. So this is an example of the emission of DAPI, which is a very popular DNA stain or DNA dye. And if you use it together with something like fluorescein or Fluorophore 488, because DAPI has such a broad emission shoulder, if you excite and detect both of these simultaneously, you will definitely get some blue signal that will show up in your green channel, so some false positives. So that's something that to be concerned, especially if you do multiple colors and look at co-localization between different proteins. So when you're planning an experiment, you definitely should think hard about colors. What dyes are you going to use? This is a kind of an unfortunate combination here, because you have the emission in green, but then that emission will definitely leak into your next channel. So you would be much better off to choose a dye that's further apart from your Alexa Fluorophore 488. So that'll make your life much easier. So a little bit of planning will make it more straightforward to do the experiment. So let's talk about objectives. Because now the most common question that I get on when someone just wants to know a little bit about microscopy, I show them the microscopes, and then they will ask me, well, what's the biggest magnification that you can do? And my answer is, it's unlimited. We can magnify 1,000,000 times. But just on a little bit more serious note, when you look at the microscope objective, this happens to be a Nikon, but it would be similar in any other objective. You have lots of information on it. Now, the most important number on this objective is not the magnification. Really, the most important number is this number 1.4 on this particular objective. That's called the numerical aperture. I'll talk about it in a little bit. And that should really be in giant numbers because that is what determines resolution. But okay, we do have the magnification here. We also have this number, 0.17. That's also important because it tells you that it is designed to look through a cover glass of this thickness. So if you use a thinner cover glass or a thicker cover glass, you cannot get as good an image as if you pick the right one. And then this here tells you that this objective is corrected for flat field, and it's also corrected for chromatic aberration. Again, I'll talk about it in a bit, but the short of it is that the numerical aperture is really something that you have to think about, the most important number. So what is it? If you take two different objectives, and let's take this one, and this is the focal plane, and really the numerical aperture describes how wide a cone of light that is coming out from the sample, how wide a cone the objective can collect. So in this case, we have a narrow cone or a much wider cone of light. So if I had a fluorescent particle here, this higher NA objective would collect many more photons. So you get a brighter signal. You also get higher resolution. So that's the number that was on that objective. And you calculate it, or they calculate it by taking the refractive index of the medium. So if it's a dry objective, a refractive index is about 1. If it's oil immersion, it's about 1.515. We also have water immersion objective and glycerol and some other. So times sine alpha, which is half of this aperture angle. So here is that again. So if you have a fluorescent dye particle and you excite it, the photons can go in any direction that are emitted. So we only collect a very small fraction of those. So the higher the numerical aperture, the bigger fraction of these photons we collect, and we get brighter signal. So it's still only about maybe 30% of all total photons, but it's better than on the lower NA lens. So we talked a little bit about magnification, and it's not the same as resolution. So in this case, we have the same magnification for two microscopes or two objectives, except the one here in the bottom also provides a much higher resolution image. So that's why I was saying we can magnify as much as we want, but the resolution is limited by laws of physics in most cases. So at the core is diffraction, and essentially what you could or should know maybe is that the image of a small point object is not a point, but actually an airy disk or airy pattern. You can see it here. So it's a central bright disk, and that's surrounded by these concentric lighter and darker fringes. So if you take a series of beads of decreasing physical size, when they're fairly big, then the size in their size in the image will correspond to the size on the slide. But once you hit certain limit, the diffraction limit, even with smaller and smaller beads, the size of that spot, of that pattern will remain the same. That's your airy disk. Here is a little bit more info here in this link down in the bottom. And it's called the Airy disk after Sir Airy, who was the royal astronomer, because astronomers like to look at small point objects like stars. So now that we know what an Airy disk is, or Airy pattern, now we can talk about resolution, because resolution tells us how close can be those two Airy disks or Airy patterns be together, and we can still tell that it's two and not just one. And there is a formula for that, one of the formulas, but this one is used fairly often. And it's 0.61 times the wavelength of light divided by numerical aperture. So as you can see, there is nothing about magnification of the objective, because that's not really what determines resolution. And this formula is called Rayleigh formula, after Lord Rayleigh, and who came up with this definition. So one thing to remember, if you have a small number, means higher resolution. So if you have two objectives, one has a resolution of 0.5 microns and the other has resolution of 0.8 microns, the 0.5 is a higher resolution, so don't get confused. Similar thing goes for a Z resolution. The formula is more complicated, so don't worry about it. But this just shows that a point object actually is represented by the optics as a three-dimensional pattern. So if we look along Z, we can also get the central bright spot, and then we have this first minimum. So we can measure either the distance from the center to the first minimum, or we can just plot the intensity and then find half of the maximum intensity and measure how wide the peak is. And that's called full width at half maximum, so you can sometimes see it. You can also see that between different numerical apertures, the length of the Z resolution is very different, because the resolution increases with the square of numerical aperture. So if I go from 0.5 to 1.4, that's almost 3 times higher, and I will get about 9 times higher or better Z resolution, meaning smaller number. So now we know all about resolution, we know about Airy disks, and so let's look at some practical matters. So what the relation between brightness and numerical aperture and magnification. So essentially what you need to remember from this is that you would want to use the highest numerical aperture objective so that you collect more photons, but also the lowest magnification objective so that you don't spread the photons over a large area on the camera, because then you would get dimmer image. So that's and here is an example of two objectives that have the same magnification but a different numerical aperture. This is a plan achromat, and you'll see what it is in a bit. The NA is 0.65, so it's probably a dry lens. And this is an old image, so it was taken on a film camera, but the exposure was about 3.8 seconds. And then if you go to the next objective, which had the NA 1.3, it was an oil immersion lens, and it only took about 600 milliseconds. So much shorter exposure. Also, that image looks much sharper. So we assume that it's got better resolution as well. So objectives, numerical aperture, and such. So what is the best objective? Well, the best is the one that's suited for your sample. It's like asking, what is the best car, a Lamborghini or a Dodge pickup truck? It depends what you need to do. So essentially you only get good images if your sample is what the objective was designed for. And so because what complicates matters are some aberrations. So for instance, well, I'll show you on the next slide here. So one is spherical, and that essentially means that you cannot focus all of that light into a nice, tight, small spot like an airy disk. Instead, you get this fuzzy, blurry spot. And so that makes, that degrades your resolution. The other problem that you can have is chromatic aberration, where different wavelength of light or different colors of light have different focal length. So even if the object, your sample, has a small particle with that emits red as well as green or blue, those will not necessarily end up in the same plane in the image. So colocalization would be tricky. Now this has been known for a long time, and the objective manufacturers came up with different designs to correct some or most of these problems. So you can have a series from low-end achromats to fluorides, the apochromat lenses, which essentially have very small chromatic aberration and also are typically well designed so that they minimize the spherical aberration. Here is an example of some of these objectives. You can see that the better corrected, the more lenses or groups of lenses had to be used. And so those Apple lenses are also the most expensive. So the practical limits of resolution is your aberrations, whether it's because the objective is not totally corrected, or you are using the objective for a sample that's not suitable for it. Of course, we also have noise from the cameras and noise from the sample, and the fact that sometimes the image is either too dark or too bright to be able to resolve anything. So really, what are, so spherical aberration, I'll talk about it a lot, so I apologize. But it's really the most serious problem, and most of the time, it's really you as the microscopist who is to blame, or at least, in many cases. For instance, if you randomly grab a box of cover slips and you don't really care whether it's how thick it is and put it on your slide and expect that you're going to get the highest resolution image from it, it may not necessarily be the case. Yeah, so most objectives are designed for 0.17 millimeter thickness of cover glass. And then, of course, if your sample itself has the wrong refractive index, then especially if your sample is thick and you need to focus deep into the sample, you will not be able to get a good image. Sometimes, of course, the sample itself is problematic because it's variable, it's non-homogeneous, and so you can have cell nuclei that act as little lenses, or you can have some other organelles, starch grains in plants is very bad. And so the idea is then that you could remedy that if you extract as many components out of the sample, but still leave the important parts that you're interested in, and infiltrate that sample with some high refractive index compounds that will make everything very optically homogeneous. So it could be glycerol, methyl salicylate, some synthetic resins, but you cannot do this to live cells. So let's look at a cover glass thickness. So number 1.5 cover glass corresponds roughly to the right thickness. Some people take calipers out and they actually measure each cover slip if they do something really high resolution that's critical. Or you can pay more money and you get these specially selected cover slips that are guaranteed to be within about 5 microns from the right thickness. So typically that's important for your high numerical aperture dry objectives and also for water immersion objectives. So here is an example, an image taken with the correct cover glass for which the objective is designed, and then another similar sample where the wrong too thick cover glass was used. And you lost a lot of resolution there. Okay, so practically speaking, if you use dry objectives, so they worked fairly well, but mostly for thin samples, maybe 10, 20 microns thick. It depends, what their numerical aperture is and resolution. The higher the numerical aperture, the more sensitive they will be. But of course, if you have samples that are mounted in something like glycerol or some of those commercial mounting media, they will have probably a refractive index around 1.45, 46. And for that, you may use an oil immersion lens, but it's not a perfect match. Ideally, you would want to use a glycerol immersion objective. Or if you have a microscope with oil immersion lenses, then you'll find a mounting medium that has the right refractive index. You can buy them nowadays. Like for instance, there are many, but for instance, Thermo Fisher sells something called prolonged glass that has the right refractive index. Citifluor is another company, and there are lots of others, or you can make some of your own. Some media remain liquid, so you have to seal the cover glass so that it doesn't slide off or dry up. Some other media are hardening media where you put the drop on it and eventually it'll kind of dry up, but then it also tends to shrink. So if you're looking at some 3D images, you may get quite a bit of distortion. So a lot of these mounting media also will have those antioxidants or anti-fade reagents. And you just have to be careful because certain reagents are not compatible with certain fluorescent dyes. So, read the fine print. Okay, so that's the anti-fade. I think I'll just skip that. You can, look at the options. So, okay, so we have the sample prepared. We pick the right objective. And you're looking at the microscope, and then the question is, how do you record the image? Of course, on a camera, on a digital camera. And there's a couple different kinds, but and you'll see them on the next slide once I show it to you. But typically, you have either front illuminated cameras or back illuminated. So on the front illuminated camera, you have the silicon substrate, and then you have some photodiode arrays and some circuits and everything else. So the incoming photons, they have to get, some don't give you any signal because they did not hit the photodiode. There are all of these circuits that they have to go through. And so their typical efficiency could be, say, from about 40 to about maybe 70%. So out of if you shoot 100 photons, between 40 and 70, depending on the camera, will actually result in some sort of a signal. The rest has been lost. So that's why some other cameras are back illuminated. This is where they took that same chip and then they thinned it and then turned it around so the photons are coming through that now really thin silicon substrate. And that gives you a really high quantum efficiency. So 95%. So out of 100 photons, almost every photon will give you some sort of a signal. But that also costs a lot of money in most cases. So these cameras could be really expensive. So what cameras are there? In general terms, the most sensitive and also the most expensive would be your electron multiplying CCD cameras, or EMCCDs, and typically back illuminated, and therefore best for extremely dim signals, let's say less than 10 or less than 5 photons per pixel there. Yeah, but once the signal is brighter, you actually get a less noisy images from other cameras. So the other, nowadays, the most popular choice is the scientific CMOS camera, which have fairly low noise, large chips, and they can be really fast. And those are now most popular. So if you buy a new microscope and it comes with a relatively expensive camera, this is what will be on it. In previous years, the CCD cameras were what was typically used, but nowadays they have been mostly replaced. Now, sometimes you don't want to spend $15,000 on this scientific CMOS, and there are many just regular CMOS cameras, mostly used for like machine vision or video surveillance and such. And those are typically much lower cost. So they have a little higher noise, but for many microscope applications, they work really well. So instead of a $12,000 camera, you can get a $500 camera that works almost as good. So that's, so how about color? Well, that's a good question, because we have fluorescents that have different colors. And so, but you typically don't have to, or probably don't want to use a color camera to image your fluorescent signals in most cases. So to get color images, you could do a couple of things. You could take 3 images with different filters, the red, green, and blue filter with a monochrome camera, and then the software will put those together and you have a color image. Or what is most common, the chip itself has a mask Bayer filter or similar, where some pixels only see red, some only green. Typically there are twice as many green pixels as the other. Some only see blue. And then the software afterwards has to kind of average and put together or interpolate the location of your red signal or blue signal or green signal. You can see already that if you're looking at a red signal, you're throwing away about 75% of the signal, because the other 3 pixels don't detect any red. And that's similar for the blue, and to some degree to green. So color cameras are typically less sensitive, have lower resolution because you have to interpolate those pixels. And so in majority of cases, you're much better off with a good monochrome camera and some good fluorescent filters than trying to do color. Because essentially to the camera, it doesn't really matter what color it is. We know that these were green, this was green color or blue or orange, and we can always assign a pseudo color to it afterwards just for presentation. That's how that's typically done. Okay, so for this section, some take-home message or a recap. Typically, you will want your objective immersion medium, say oil immersion or water immersion, to be closely matched by the refractive index of your sample. So if you have a live sample that's mostly buffer or water, you should use a water immersion objective. Or if you have a fixed sample mounted in some high index medium, oil immersion is probably going to be reasonably good. And Yeah, you want to make sure that the sample doesn't move, so vibrations or the sample moving, that's typically a bad thing. Use high NA objective, as high as you can, and low magnification if you can, so that you get a really bright image. And of course, pick your bright fluorescent dye or stable bright fluorescent proteins. It's use only as little excitation as possible and short exposure time so that you don't photo bleach your sample. So now let's talk about noise. So what is signal to noise? So your signal has to be discernible or detectable above the background of noise. So here is a picture I took earlier this week. And this is a plant leaf expressing fluorescent protein fusion here with 10 millisecond exposure and really low excitation light. You can see the signal, but it's barely discernible above the noise background. I mean, if you look through the eyepieces, you don't see anything. It seems to your eye, it seems totally dark. Now with adding a little bit more light and increasing the exposure, now we get a much cleaner image with the higher signal to background noise. And whether any particular molecule emits a photon or not is a random process, and we talk about statistical noise or Poisson noise. And that noise is independent of the hardware. Of course, hardware will cause other noise like thermal noise. That's why we have cooled cameras or readout noise. But the statistical or Poisson noise is what we'll address here. It's named after this person who was a French mathematician and physicist. Okay, so what is that noise? So that noise you calculated as the square root of the number of photons detected. So let's say if you on average detect 16 photons, then the square root is 4. So you have about 2/3 of the cases. Let's say you took 100 images, and it's 66 of these 100 images, the brightness or you would be detecting somewhere between 12 and 20 photons. But in the other 33 or 34 cases, you would actually detect more or less than that. Even though everything else is the same, it's just the random nature of that photon flux. And the fewer photons you have, the higher the effect of that. If you only detect 4 photons on the average, the noise is 2. So in 2/3 of the time, you'll detect between 2 and 6 photons. And in the other times, you will detect either less or more. So really, it's a hard limit. And the only way to get around it is just to collect more photons. That's why I keep mentioning numerical aperture is really important. The more photons you collect, the better off you are. So only if you collect enough photons, you can be certain that you can resolve certain features in the sample. And so you either have to take longer exposures or use more excitation light or average number of images. And so every photon lost in the microscope is going to cost you. So try to minimize those losses. So that's the noise, and then there's a little blurb on software, but maybe we don't really have to talk about it too much because, but maybe just what I wanted to mention, if you buy a microscope, typically comes with your control software for people who build their own microscopes. They probably write some code, but there is also this, this is a great piece of software, the Micromanager, which can control lots of microscopes and cameras and other hardware. And you can give that a try because it's free. And as far as analysis, I like to start with ImageJ or Fiji because it's free and it can do a lot of things. Before you get to the point where you need more sophisticated analysis or maybe some 3D analysis that ImageJ maybe cannot do right away. So that's the really brief intro into fluorescence microscopy. So that's that. Now, I'm pretty sure that there will be lots of questions. So I'm happy to take questions, answer your specific complaints if you have any. That will be. And then if anybody is interested, you can hang on, and I'll walk over to our Leica fluorescent microscope, and maybe we'll share a screen and look at a sample, and you'll be able to see, the noise and maybe some parameters that I'm changing to minimize it. So that's. I think that concludes this this little show. So,
thank you, Stan. Are there any questions for Stan before we move on to the demonstration?
there gotta be questions. I hope there will be questions. Well,
you did such a good job. No one has any questions.
It's a very quiet group. That's always very suspicious. Oh, I don't know. Well, I guess what I'll do, if you cannot think of a question or don't want to ask before anybody else, you can shoot me an email or call me or just visit the MIC. If you can get in, I guess we can arrange to open the door for you. Or if maybe you have a microscope in your lab that you're not really sure what's wrong with it or what it can do, I'm always happy to talk about microscopes and cameras and such. So let me know. Okay, last 5 seconds to come up with a question before I go to the other microscope. 5, 4, 3, 2, 1. All right, well, I guess I'll walk over there and you'll see me again in about 3 minutes if there's anybody left here in the group, okay?
Okay, I'm gonna make that other one a host.
Okay, I'm gonna stop my video. Actually, I think I'll just log out of this one.
Okay.
All right, I shall see you guys later. Hello again.
Okay, I think you should be the host over there now.
All right. Yes, I am. Okay. So let me show you. This is our little Leica microscope, and we have a monochrome camera and a color camera, but we'll be playing with the monochrome. And I put a slide on it. I'm not really sure exactly what it shows, but we shall see. And so what I'll try to do actually is share a screen that shows the Leica software interface. There we go. And so, all right. So I don't know if you guys can see it. I'm gonna turn the font a little bigger.
We can see it pretty well.
Okay. So this camera, I will just switch it here to low noise, which means high sensitivity. And let's see if I can get any signal on there. Well, I guess I can. Let me turn the room lights off and make it a little more in focus. And so there is an example of, I'll show you an example how the numerical aperture and magnification play together. Because currently I have a 20x objective, dry lens, with numerical aperture about 0.55. Okay, and then I have when I will switch to and it's a reasonably bright image. Here it's in black and white, but let me give it a fake color. And let's say that this is, let's just call it green. All right, so when I turn the light on again, oh, now it's green. Yeah, but I can also show it in the pseudo color, and the blue tells me, oh no, watch out, it's too bright. This will be saturated. Yeah, so maybe I'll turn the exposure down a little bit. Okay, all right. So this is the 20x, 0.55. I have a 40x, which is only slightly higher than 0.6, so essentially the same. And when I switch over, you'll see that it's much dimmer. Yeah. So for the little extra resolution, the 0.55 NA versus 0.6, I paid a lot in the number of photons per pixel. So much dimmer image, and didn't really gain that much resolution. Mostly we like this objective because it is designed to look through a fairly thick layer of sample or a glass window and such. But otherwise not so great in terms of resolution. So I'm gonna go back to the 20. And I could go back to our 10x objective. All right. Yeah, that is a little dimmer because it has lower NA. But I think it shows still okay. So, is anybody there left who actually has questions? Okay. Oh, maybe. I guess couple of people are still hanging on, so and on this particular microscope we could of course do multi-color fluorescence. So for instance, I could add, because it's most of it is motorized, at least the filters. So I could add another channel, and here in this channel, which I'll label red, if it lets me, label it red, and I will pick the Psi-5, which is this near-infrared dye, and let's see if I'm going to see any signal. That too shows, pretty much very similar signal. So I don't know if my sample is just not terribly specifically stained or not, but let's see if we can find something that's more distinct. No, maybe not. So maybe we can try something like GFP filter. Oh, that is bright. Okay, so I guess I could have picked a little different sample to impress my audience. Yeah, that is still pretty bright. So I'm gonna go down and turn the intensity. So this microscope has LED light source. So I can change the brightness of the source quite easily. It'll be harder on the, if it was the old-fashioned marker one. So one thing is, which I really like about this, this microscope is that we can, let me just do one channel, we can do stitching of multiple areas on the microscope. It's one of my favorite features. So this is called the navigator, and here I can go, again, this is the live view that shows, and I say, oh, let's go around in the spiral and see what all is here. And it's going to go around. I'm gonna zoom out so that we can see it a little better. And yeah, it would be going around and around in the spiral, but maybe what I could do instead would be to say, well, let's see what's over here. And I'm just gonna click here and see if there's no sample there. How about here? Okay, there was still something. So I can do a little exploration. And if I feel like I found most of the sample, then I'll say, oh, let's just image this whole thing. Like that. It's probably not the whole thing. But here it tells me that it will be 18 slices. And I can, for now, I'll just do a little preview. And it's going to image all of these tiles, and then if I was doing the actual acquisition, it'll stitch it all together into one giant image. So this is fairly convenient if you're looking at the large tissue sections that you need the image at a reasonably high resolution. So this was fairly quick. Sometimes you have a really big sample and you need to use a higher resolution objective. Then you end up taking several hundred of these tiles, and your images are really, really big. But that's the navigator portion of the software. Pretty handy. And of course, let's just find maybe let's go to live view again, and I'm going to show it 100% on the screen. And this is a fairly flat section, so it'll be a little, I don't know that it really allows me to demonstrate the 3D imaging, but we could, you know, if we wanted, we could take a stack of images, you know, a Z-stack, to get some sort of a three-dimensional representation of the sample. But that's typically, the sample would have to be much thicker for it to make sense. Or we would have to use a higher resolution objective, maybe the oil immersion, so that you know, we get better Z resolution. So that's fluorescence in its full glory. Well, I think if there are questions, let's see if anybody has any burning question or issues. If not, well, just let me know when you do have them, and I'll try to answer them as best as I can. Thanks for coming.
Thanks, Dan.
You probably cannot even see me because it's so dark.
You are in the dark.
All right.
Any more questions? Last chance.
They know everything already.
Yeah, they're so smart.
Okay, cool. I'll see you guys next month, or I think there will be another lunch and learn before you know it.
Mm-hmm.
Okay.
Thanks, Dan.
Okay. Bye bye.